I am Ian Anderson. I am Associate Director within Analytical Sciences at Pharmaron Gene Therapy here in Liverpool. We offer a fully integrated end-to-end gene therapy AAV platform service which starts with both development and manufacturing capabilities. We have advanced analytical platforms and high throughput technologies that we can use for process development, and we have both GMP manufacturing facilities to allow us to make adeno-associated virus to GMP, but also to make plasma manufacturing to GMP as well.
My name is John Allen. I'm the Senior Manager of Analytical Core Technologies here at Pharmaron. So we're primarily responsible for method development and the validation of HPLC methods to support AAV platform analytics. So Waters use PLC instruments are the cornerstone of our analytics really for AAV platforms in supporting our customers, because we use them to analyse the critical quality attributes which are crucial for providing best in class of the product, for the final product for the customer. So this can range from looking at the titer of the AAV, we're also looking at the purity, so titer by reverse phase, purity, it could be SEC, looking at aggregation, or it could also be more residual analysis, which we'll touch on a bit later. And there's also looking at the empty full analysis, which is also crucial, which we do by ion exchange.
We've developed an anion exchange method for each of our AAV serotypes that provides us a high throughput empty full analysis of the product to allow fraction screening during the process as part of our process analytical technology. In addition to that, we have size exclusion chromatography for the characterization and aggregation assessment of our compounds, allowing us to perform stability studies as well as release testing of AAV compounds.
So all our LCs are coupled to an mPower network, which we find such ease of data transfer and sharing, and also it's a powerful tool for analytics, and it basically makes it fastest way to produce our results to the customer.
Using the Waters UPLC and the great service that comes with it, we minimize the downtime of our instruments, as well as allowing us to perform critical analysis with low sample volumes without risk of malfunction.
So residuals come with this wider bracket of impurities, so these can be product related or process related. So we have a plethora of methods to analyse this, and why they're important is they could be either toxic to the final products, or they can affect the process. So we have methods that look at the in process and also the final product. So traditionally historically we used to actually outsource this to other companies because we didn't have the kit on site, but 3 or 4 years ago we actually purchased additional detectors for our existing Waters kit, because typically these residuals they have poor UV absorbance or no UV absorbance, so you have to be a bit clever in how you analyse it, and the Waters kit has an array of different detection methods and flow cells which allows us to do this basically. So for examples for the residual methods we have, a good one we have at the moment is Pluronic. We tend to use the C18 technology, but this one uses mixed mode. It allows us to separate the sample matrix of the AAV and the AAV from the product. And we use ELSD because it has no UV absorbance, so you couldn't do that on any other piece of kit out there. It allows us to have very sensitive, and it allows us a quick method. It uses the switch valves to remove the impurities and uses the hydrophobic properties of Pluronic to allow us to assess that. We have that for in-process and also for the final product as well. We also have a residual and a titer method for that. Other examples include Triton X100, which is used to lyse the cells. We've also got PEI, which is used as a transfection agent. For the PEI, for example, we use the fluorescence detectors on the Waters instrument. We find this to be very sensitive and also a very good method when we've labelled it in comparison to others. And in terms of the Triton X100, we also have anti-foam, and these are poor, if not no UV absorbance, so we have to be clever how we assess those. And we also have TUV detectors and PDA detectors, and we found the TUV, which you don't get on other products, it removes noise and it finds more sensitivity. So we're basically utilising the hydrophobic properties of these molecules to tease them out of the sample matrix in order for us to analyse them and produce a product as safe as it can be for the customer.
Our analytical quality by design experts develop our methods in an agile development environment using the mPower 3 network, which is fully compliant to 21 CFR Part 11. This allows us to transfer our development methods, which have been pre-qualified, to our quality control department in our cGMP laboratories for final validation and release testing.
So in conclusion, the Waters instrument we find to be very robust, very reliable, very sensitive, and it gives you an array of detection methods coupled with the mPower network. It makes it a very fast delivery system for the final results and allows us to produce best in class and work with our clients to produce that.